Okay, so welcome to the afternoon session of this very interesting meeting. So I'm Apratim Chatterjee from ICER Pune, and I'll be chairing the session, the afternoon session. The afternoon session has all modeling and computational talks. So while the bell will ring at 20 minutes and the talk shall finish in 25 minutes, what I think is if there are very brief clarification questions in the middle, in case because it's a very different modeling talk, let there be some one or two very short clarification questions even as that talk goes on, because all are modeling talks. With that, I will just invite Professor Daniel Jost from ENS Lyon to give his talk.
Thank you. Can you hear me in the back? Yes, cool. Okay, so thanks Apratim. So first I would like to thank, of course, Dimple and Ranjith. Mithun is not there, so I will not thank him. So anyway, so thanks a lot for the organization, for the invitation. Thanks also to all the volunteers that are here at ICTS. Also, it's a very wonderful meeting, and it's my first time in India. I really love it, and hope to come back more time. So I will present today my work, or our work actually, on the role of polymerases in driving the 4D genome. So my group in general is interested in the structure-function coupling of chromatin, and basically we usually have two types of questions. So how the basics, biological processes that are occurring at the chromatin scale may shape the chromatin organization and dynamics, and reversely how this organization may have an impact on these biological processes. So of course, a lot of nice feedback between these two processes. And so to do that, we are usually biophysicists in the group, and so we use biophysical modeling in collaboration with experimentalists, but also sometimes we look at the data we're around, and we make our own analysis to try to make the connection between mechanisms and experiment. And so the two next slides are two kind of teasers. So you all, we already have discussed about different mechanisms that are driving the 3D genome organization. One of the very important, I think we believe that it's very important one, is loop extrusion mechanisms that are driven by SMC proteins like cohesin and condensin. And so my teasers will be this one, this paper that just was posted 2 days ago. It's a nice collaboration with Forel Piazza, a colleague and friend at my institute, where we look at loop extrusion of condensin in yeast. It's a very nice work. I would be happy to talk about it if you want. Another very big mechanism that drives the large-scale organization of chromatin is a phase separation-like mechanism that are driven by architectural proteins. And again, we have a long history in looking at these questions since 10 years now. And again, a small teaser for papers that appeared last year. Very cool collaboration with Gary Karpen, where we were interested more specifically to the formation of HP1 condensate. Okay, but today I will focus on a slightly different subject. So we have these two main processes. I mean, one is likely related to translocation of motors along the DNA, okay, has the way loop extrusion is working, and somehow we have also a mechanism that leads to condensate and likely driven by attractions between molecules. So, I mean, as a theoretical physicist, I think a question that we ask is, what happens if you now kind of mix these two types of processes, translocating motors with attraction or with condensation? And actually in the cell, in the nucleus, there are two well-known motors that are translocating that potentially have this capacity to attract each other, which are polymerases. And so RNA polymerase is associated with transcription, and DNA polymerase is associated with replication. They have both been observed to form kind of condensate, okay? There might be some controversy depending on the resolution of the microscope you use, but there is this notion that you form factories, transcription factories, replication factories, which is basically assembling at the same place different transcription units and different replication units. And so the question we wanted to ask is how this potentially phase-separating self-interacting molecules, which are translocating, may potentially shape the 4D genome. And so I will start by discussing the DNA polymerase problems. It's highly likely that I will not have the time to talk about the second one. So again, I'm very happy to discuss. And I start with that because it's a very recent work that was just accepted. And also because in the audience, there are people that have already heard this story and not the other one. Anyway, they are both very interesting. So how replication may reshape the genome organization during S phase. So at the very large scale, I mean, again, back to this type of picture where people have observed that component of the replication machinery potentially form clusters. And this is related to this idea that the forks that are working on DNA may form condensate. This is whole picture. So more than 10 years ago, using still SIM, so super resolution microscopy. So they do observe that it's in yeast. But along the years, there have been more controversy about the reality of this condensate that could just be a resolution artifact of the, and because people were using a super resolution microscopy, so this is in mammals, where you use just normal confocal, you see condensate, but when you now use super resolution microscopy, you do see smaller condensate. Basically, you go at the fork replication scale, and so this questions the reality of this condensate, which are just condensate because they are just forks that are just close by along the genome, and that's it. Nothing really, no real forces that are driving this condensate formation. So these are at the large scale. At this, let's say, shorter scale, there are also evidence that sister forks, so basically when you, at an origin, you start a bubble of replication and you have replication in both directions, there are two forks that are called sister forks that will go in opposite directions. There might be some evidence that these sister forks may also colocalize in space, okay? Remain bound together. And there are evidence potentially in old evidence by the Tanaka group in yeast. I will discuss that experiment later. And there are potentially also molecular evidence that some proteins, like CTCF4, might be the regulator of these interactions between sister forks. So there is this different degree of association locally and at large scale. So we wanted to a little bit make rationalize this type of interactions, try to see if by building a quantitative model that integrates these ingredients, we can now look at the data with this kind of mechanistic eye. So to do that, we first have to account for replication and to introduce the replication dynamics. And so to do that, we use very basic kinetic models where the replication is modeled very classically somehow. So you have origins of replications that can fire. So this will open a bubble. And you have sister forks that along the genome will progress in opposite directions, increasing the bubble size. You may have another origin that is fire just after. You will have another bubbles. You have progression. And then these bubbles, when they meet, they merge into higher bubbles, etc. So you have the replication of the genome that is like. So in eukaryotes, you have multiple origins. In bacteria, you usually have one origin. And we will mostly focus in eukaryotes. We have this very simple dynamics. So we implemented a kind of stochastic model for that. And the idea would be to take this 1D dynamics and to put in 3D in a polymer that is moving, okay? And so what you are seeing here is a kind of GIF animation of simulation where we simulate the chromatin in 3 dimensions, and on the same time you have these dynamics of origin firing, spreading of the bubble, merging, etc. And so basically we explicitly account in the model of the formation of an extra chromatids. Okay, so these red and blue pieces are the 2 sister chromatids that are produced. Okay, so we have a framework that allows us to try to look at what's the effect of having replication, and on top of that, having two chains that are emerging from the duplication. Okay. So basically, rapidly, I mean, first of all, it's quite an unusual physical object because you have a kind of out-of-equilibrium process, the replication, and at the same time, you have the dynamics of the polymer. And so, as a physicist, it's always interesting to have this kind of weird beast in our hands. And basically, for the sake of shortening the description, we will mostly focus on the talk on two hypotheses, going back to this first observation that potentially sister forks may interact together. So we just scan two scenarios, for example, and we look at the behavior of this object if basically the two forks have no interactions, they just move independently from each other, or they are bound together by CTCF4, for example. So it's just my only physics-like picture, at least with strange names that some of the biologists will not care too much. So here you have, since it's an out of equilibrium process, you have an interesting coupling between the dynamics of progression of the replication forks and the dynamics of relaxation of the polymer. As you go for very fast replication or to very slow replication, the behavior are slightly different. I will focus here on the physiological speed, okay? And actually at physiological speed, sorry, you are in the regime where the progression of the replication is slow compared to the polymer relaxation, okay? So at a given time, if you have a distribution of loops like that, the polymer somehow has a time to equilibrate between you have a significant change in the loop topologies. So you can show that basically all the 3D dimensional properties are the one of basically a loopy polymer like that. And in the case where you do have interactions between sister forks, you form this kind of weird system. Here we have loops also. Actually, you can make a very easy parallel between this type of process and the loop extrusion process, okay? Because basically the forks will still progress in the opposite direction to replicate the DNA, but they will be stick together and you will be extruding loops, the nascent loops from the sister chromatin from that. So it's a kind of loop extrusion mechanism here. And you can make a parallel with the polymer bottle brush, but I will not enter into too much detail here. Okay, so now we have this framework. We can compute some different prediction about the different scenarios, and we wanted to compare with the available data sets that are on the market. So the first data set, it's basically related to this Tanaka paper where they were suggesting that you have these sister fork interactions. So the experimental system is very simple. So they color two loci, these two ones, and the particularity is that they are symmetrically positioned around an origin of replication, a strong origin of replication. So basically these two dots will be replicated at the same time, in average. So as the application is progressing, these two dots will be replicating at the same time. And so now what we can do with the simulation is that we can monitor as a function of time what is the typical distance between these two pairs of loci in one of the two scenarios. So that's the two types of prediction. So for example, where the two sister forks move independently from each other, and you monitor the change in distance. So basically, before these two loci are replicated, you have a given distance. This distance shorten a little bit and then increase. The times where this distance is shorter is basically the times where the 2 points are replicated at the same time. And why you have a slight decrease in distance is basically at this time, you have a loop, and the two sides of the loop are the 2 points where we are monitoring the distance. And so if you know a little bit about polymers and ring polymers, it's not very surprising that a polymer rings is more compact than a linear polymer. And so that's why you have this slight decrease in distance because the object in between is more compact. Okay, so you still have this slight decrease. But now, of course, if you introduce the pairing between the sister fork, you have a huge and massive depth in the distance because when they are replicating at the same times, they are more or less occupy the same space in 3D dimensions. So you have this huge decrease. Sorry. And so what Tanaka was measuring is the blue bar. So the statistics is not fantastic, so you cannot really make a statistical test. You can somehow discuss the strategies they use to analyze the data. Anyway, that's the only thing we have in our hands. But with this, you can see that, and that's why also they make their conclusion in the paper, so it's fully consistent, that it's very consistent with this extrusion-like mechanism with interaction between sister forks. It's not fully compatible, so likely this picture, the experimental picture, is compatible with a mixture scenario where you have the majority of case extrusion and some case where you have independent fork progression. So that was a first test of the things. But now, okay, we have also access to high-C data, which potentially have enormous capacity, enormous statistical power because there is many loci that you can look at the same time. In microscopy, usually it's loci by loci that you are looking at. It's very. And in high-C, what you would expect, okay, from this type of scenario. So, for example, for the loop extruding scenario, what you would expect around an origin of replication. So, let's say in the middle you have the origin of replication. You fire a bubble, and so the bubble will symmetrically progress. And so, what you would expect is that the loop on a kind of fake IC map will move along the counter diagonal of the map, okay? Because you remain symmetric regarding the origins, okay? And so that's basically the high-C prediction of this with more statistics and looking at a little bit of stochasticity and heterogeneity in the system. So this is in the sister fork interaction system. This is in the no interaction system. And you see that as a function of time, you have the kind of fontaine or jets that are appearing, that are propagating because the replication bubble is increasing, and then that's fading when your replication is done. While in the case where there is no replication, you have slight signals here, but it's not very clear. So with the model, we basically, the model helps us to make a quantitative prediction is that around origins of replication, you should potentially see this type of object, or not, if the loop extruding like mechanism is not really happening. So we again focus in yeast because in yeast you have a lot of high-C data in S phase, which is not really the case in other organisms. And to do that, we somehow go larger and not focus on just one chromosome, but we aim to also look at the global organization of this replication process. So we build our own polymer model of yeast organization. So you may know that yeast have a very Rabel organization where all the centromeres are bound to the spindle pole body at one pole of the cells. Telomeres are attached to the membrane, and one part of the nucleus is occupied by the nucleolus. So this is a very schematical view of that. So this is an example of our simulation. This is just to show that with these very simple ingredients, it's just very simple homopolymer model that are just constrained, centromeres to the SPB, telomeres to the membrane, and just with that, you can get a very nice, not fully quantitative everywhere, but quite nice picture of what's going on in the experimental high-C. So that's for the 3D organization and also for the 1D replication dynamics, because we wanted to also reproduce the correct one. We tune a little bit the probability to fired origins, the speed of progression of forks, in order to recapitulate the mean replication times that have been measured experimentally. So this is for the 16th chromosome of yeast. In black, this is the mean replication time as a function of the genomic distance. And in blue, this is our stochastic model of origin firing and progression that nicely mimic the mean replication time experimentally measured. And basically, the idea is, of course, to couple the two things and see what's going on in 3D. So here it's, for example, an example of a simulation where you have the monomers that are in white here. The green part are the forks. And you can see the red and blue, as before, are the two sister chromatin that are actually formed. And then you can track the dynamics in the 3D space and basically compute high-C maps. There is a lot of work to do to understand how to compute a good high-C map in S phase, because in S phase you have different copy numbers depending on where you are, depending if the region have been replicated or not. You have two copies or one copies. So there is a little bit of data analysis to do. We did it. And you can now compute what should be the high-C map around origins as a function of time. And so this is the case of the loop extruding scenarios when you have interactions. And you can see that you have the formation of these fountains that is growing, and I didn't show, but at the end, it just disappeared at the end of replication. All right, so we can now, with this framework, predict this type of patterns. Okay. Do we see this pattern in the data? Yes. And so that was, I think, to my knowledge, the first time here that people have observed these patterns related to replication. So with our colleague, Aurel, he's a yeast guy, he's a high-C guy, so we asked him, okay, just do for us a high-C map in early S. So he synchronized cells in G1, then release for a short time, and then fix the cells and measure the high-C map. And then when we pile up the high-C map around the origins, we can nicely see these fountain-like patterns that are emerging from the high-C. You do not observe that in G1, and you do not observe that in G2. So this is really specific to S phase. Okay, but there is some subtlety in yeast that you may not know, is that cohesin is there, and is there, it starts to be there, and start to loop extrude chromatin in S phase also. And also in G2, but not in G1. So one may argue that actually what you see is cohesin dependent. It's something that is nothing to do with replication. It's just that during replication, cohesin is also loaded and loop extruding. And so what you see is something like related to loop extrusion, but by cohesin, not by the replication forks. So Aurel, and actually it's Vinciane in his group that did the experiment. She depleted cohesin, and basically, by depletion of cohesin, you still maintain this nice fountain patterns while you are removing totally the loops that are cohesin mediated on the high-C map. We can show also by basically avoiding the replication to happen. So there is a mutant, the nice thing in yeast, there is a mutant where you can basically, the cells are running totally perfectly in the cell cycle, but they are not just replicating the DNA. And if you do that, you completely lose the fountains. So this just tells you that what you observe, this signal is really replication dependent and cohesin independent. Of course, we think that this is related to the sister fork interaction, but okay, that's the next story will be to really prove that. So now if you put the two things together, so that's the prediction at the time that corresponds to the experiment. This is the experiment. And this is when we pile up all the origins together. This is when we pile up all the origins that are far from centromeres. And you can see that at least qualitatively in the shape, this is quite representative of the experiment. It's true that this is not quantitative. So basically, our fountains are much stronger than what you observe experimentally. And again, that could be a signature of the fact that you have a mixed scenario between fountains, so replication interactions, interaction between forks, and no interactions. And so with our framework, then we can play basically, and now look at the higher order structure. So far, I was only interested in the interactions between sister forks and around the origins. But with our framework, we can now look at how the forks are dispersed in the nuclei. Do they form condensate? Here, I didn't put any forces between forks that are not from the same, emerging from the same origin, okay? So if you see condensate, it's just randomly or because of the rubble organization, this type of stuff. But even that, I mean, you can see some correspondence between the two. And so we can play a little bit with the model, so we can look at how the forks are distributed in the nucleus as a function of time. So you do observe a kind of wave that starts from the SPB, so centromeres, and that progress throughout the nucleolus. So that was also observed in the experiment. We can also look at, when we do see condensate, how many forks are there? Okay, so if we just focus on the red curves, you have a distribution of number of forks. So for example, there is many times you see 2 forks that are the 2 sister forks. Sometimes you see 4, 6, etc. In the experiments, you also see this same type of curve, but, and I forget to put the picture, you see more higher, bigger condensate. So meaning that at least in yeast, it's likely that there is some kind of interaction that drive higher order condensate. Okay, some take-home messages. So I hope I convince you that this framework is interesting to look at the mechanisms that drive chromosome organization in S phase. We show that this is a very interesting object, a physical object, but also biological object. We show that experimentally there is strong proof that the fork-fork interaction are there, and that leads to an effective extrusion mechanism, at least in yeast. There have been very recent, actually when we submit the paper, 1 or 2 weeks after, there were a paper in Nature doing a kind of version and fancy version of Hi-C to look at specifically also at this question in mammals, and they also do observe this kind of fontaines. And so, again, I want to point out that there is also nice work on the bacterial context to look at replication. So Apra team did nice work. Broders group did some nice work, and I think the next speaker will also talk about nice work about that. So it's in the air, so if you want to look at it, please have a look at this. And I will skip that just to thank the people in my group, especially Dario, who is a fantastic PhD student that did all the work. It's amazing. The funding and the small crew of people that I was with in my Zores trip. It was very nice. Thanks to all of them. It was a very nice trip. Thank you.
Thank you, Daniel. Question?
Yeah, Daniel here. So I understand your model. So, but the problem with your model is somehow you realize that the origins are in a symmetrically arranged, right, in equal distance. Because that is not true in yeast, because there is a very asymmetric division in terms of the distribution of the replication origins. So how are you going to explain that?
You mean that they are the high density close to centromeres?
Not only that, because replication origins are not really, there is no pattern in terms of how they are distributed across the chromosomes. Because some are clustered in somewhere, some are very far. So how are you going to, number one. Number two is that not all the replications, roughly around 180 to 190 origins fire at the beginning of the S phase, right? So we still don't know what happens to another 200 replication origins. So how are you going to explain that?
Basically, our model of the replication dynamics account for all these details. So we explicitly account for the distribution of origin in yeast, that is known because you have the Rs sequence. Plus, their frequency of firing is also known. I mean, we have some very good expert in my team, so Jean-Michel, who is really dedicated in his 10 last years to understand that. So he built a very nice model on that. So this is also, all this is including in the model to really account at the same time of the stochasticity of firing and the heterogeneity in origin distribution. And this, of course, has an impact on the way and these bubbles are formed, because this will have an impact on the size of these bubbles.
So just last question. So in the experiment, what you showed when you release from the alpha factor arrest, right, in the where you did the Hi-C,
yeah.
Did you use the hydroxyurea there to slow down the replication fork?
No, we didn't. I mean, with our colleague, we didn't do that. But there are work. There are people. So here I just show the data from our collaboration. but there have been all type of data already, few of them, maybe 2 or 3 other papers during S phase. They all show this. There have been papers where they use HU to treatment to slow down the replication dynamics. You do observe even nicer and small fountains when you do that. So,
okay.
yeah,
Thanks.
All the data set seems compatible with that.
Okay, thanks.
Daniel, very nice talk. So you said the fork interact, but the forks are continuously moving. So two moving location interacting with the protein looks, I mean, I don't understand how it might happen.
What do you mean? So
These are constantly, the locations are constantly changing, right?
yes, yes.
So a protein cannot hold them interacting.
So the question is what hold them together?
Yes.
Yes, that's a good question. So I think David de Maranduzo makes a hypothesis that it could be a condensate somehow of proteins that may bound them together. This could be molecular actors. So there is CTCF4 that potentially, and the picture I can show you, but the picture at the beginning showed that they potentially proteins or complexes that might be there to tie them together, like I would say cohesing rings. You may think of, even if it would not be a ring. Yeah? I don't want to say that this complex is a ring, but you may think of this as such a framework, but it's a mystery. I mean, actually, we are testing one of these actors do a mutation from one of these, but I don't have the data.
Hi. How do you incorporate the active processes of replication and extrusion? I presume this is a Brownian dynamics simulation, is it?
Yes, yes.
Yeah. So how do you impose that?
So basically, it's Monte Carlo kinetic Monte Carlo simulations.
It's a kinetic model.
Yeah.
Okay.
And so basically, with a given probability, we move the position of the forks, and then we introduce new monomers in the simulation at the position.
And these are detailed balance violating.
Yes, 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 yes. This the process of progression is detailed balance. You violate the detailed balance.
For the interest of time, we'll just move to the next.
Perfect. Thank you.